So it's my special pleasure to introduce Vicky. I actually know Vicky when she was literally a child. So when I was a graduate student at Princeton, Vicky did an internship at either a high school or undergrad in the lab. So the first figure was with me. And when I applied for jobs in Michigan, so Vicky helped me to write the personal statement, teaching statement, and research statement. And she helped me to practice the chalk talk, which I heard it was the worst ever in history. I mean, my chalk talk. So it was a truly cherishable memory. And this is something that I want to share with the graduate students here, that the PhD and when you're a student, it's really the period that you can get lifelong friendship. And after that, I moved to Michigan and Vicky moved to Princeton for her PhD study. Since then, we haven't met for 40 years. So I really want to thank Minji, Kintai, and Jane to make this happen today. I don't really go to conferences, and I don't really visit back to Princeton because I don't feel I perform well enough to do that. So, so it could be another 14 years before I see another landmate. So also I will say it's really, I feel I'm going to get a little bit emotional. Okay, so back to Vicky. After she finished her PhD at Princeton, which was focused on functional genomics, her work was on using deep learning to learn the genomic features in the genome. I think you continue the short postdoc at Princeton, the same lab, right? And then she moved to Rice as an assistant professor, focusing on integrating different omics data in understanding the different diseases, complex diseases. So let's welcome Vicky for her talk.
Yeah, thank you, Yuanfeng, for the introduction. I have to say that from my perspective, so I was an undergrad that actually was debating different fields. So I was actually econ stats major. And so Yuanfeng was also transformative in actually getting me really interested in doing computational biology. And so like a synergistic, it was very kind of serendipitous experience at Princeton. So yeah. So my talk today is on integrative computational approaches for modeling complex disease biology across scales and systems. And this is a very kind of broad title, but to kind of put things, to start things in such a, I start with a statement that I think we can all agree on. Biological systems are complex. So, what do I mean by this? And, I want to anchor our discussion a little with some of the types of biological complexity I think about. And so I actually saw that the room that I'm hanging out today has like a hard copy of this, this journal. I think a lot of our computational biologists, we anchor our this kind of area was really launched with the completion of the Human Genome Project. And people were like, oh, things are going to be solved and we're going to understand everything. When we think about the fact, if we zoom in on a single person, not to mention that the Human Genome Project was one person's genome and we have all these different people, but we think about one person, think about the fact that we have approximately around 80 different organs or tissue types. Of course, same genome, different tissues, different organs, different things happening. And now zooming in even further, thinking about just single cells, I tried to Google this number. Apparently people estimate around 35 trillion cells in like a typical adult. Now we're thinking about this complexity of like that single genome. How is it manifesting in kind of this ridiculous level of kind of scale within a single individual, right? And this is before we start talking about disease. So, now we know that disease actually, different cell types can be perturbed in different ways. I think if we really settle into this complexity a little bit, it's incredibly overwhelming. But like many other computational folks, I think, but thinking about this biology, if you just think about this, if everything were happening randomly, I think this would actually probably be a little bit hopeless. But luckily for us, genes and cell types don't operate independently or randomly, right? There's some coherent structure underlying all of this. And a lot of what we're trying to do is, if we have a lot of data, can we develop methods to mine or better model this underlying structure and learn about these different processes? So there's, I'll try to see how much I get through of this, but there are 3 computational approaches that my lab has developed recently to navigate some of this biological complexity and taking different perspectives on this problem. And so first is a method that we're calling Alpine, so guided signal decomposition of multi-batch, multi-condition, single cell expression data. And then we're going to think about actually combining single cell with another type of data, a modality, population genetics data. So the next project I hope to talk about is seismic integration of population scale disease data with single cell expression profiles. And then finally, if we have time permitting, I can talk about one of these last projects, which actually combines a little bit of my previous background and interest in looking at networks. And this project is called Etna, where I'm thinking about aligning network embeddings across species. And so jumping into this first project, so single cell this data modality has taken over a lot of biology, and people are really excited about the ability to get these high resolution measurements across a lot of different variables. So variables can be things that we're interested in, so we're calling the interesting variables conditions. So condition could be, say, different tissue types that you got your sample from, different diseases, whether a person was treated or not, the sex of the person. But of course, there are also batch effects, which have always plagued biological data, which is basically another variable that you don't care about, right? So that's what sequencing platform you use, which kit you use. And so there's been a lot of work actually thinking about how do we integrate diverse single cell data together and use it to get, and especially removing this batch effect, the stuff that we don't care about. So batch effect correction has been this incredibly active area of research. And so we were interested in is there something we can do to actually explore the conditions more? And of course, what do people currently do when you want to look at conditions? Well, one of the typical analyses people do with conditions is look at what's called compositional analysis. Simple question, when you look at different conditions, do the number of cells change? So that makes a lot of sense to look at. But of course, the most frequently done analysis downstream is to look at differential expression analysis. So basically, people actually do a lot of pairwise or exhaustive pairwise comparisons or stratified analyses. Think about as conditions explode, this is actually a very, very multi-pairwise, typically it's always control versus some treatment. And so we were interested in whether we could, directly model batch and condition in one analytical framework, instead of saying let's try to understand batch, remove it, and now do some exhaustive pairwise differential expression. And so this type of work is starting to be called, and what we call it is extracting a quote unquote disentangled representation of single cell data. So the key intuition here is that if we think about different variables, ones of interest, conditions, or ones not of interest, batch, each variable, we can think of it as having some variable specific effect that is stacking together to give us the final data set that we observe. And these variable effects could affect all of the measurements, all of the cells, or it could actually affect some cell types, but not others. But underneath it all, we do all believe that there are some inherent cell type differences that weren't affected by the variables. And when we think about it, a lot of batch effect correction or any integration method is really focusing on what's the shared similarity across the cell type. And so existing approaches that look at this, people have taken it's a relatively new area, but people have taken 2 different perspectives. One is actually using matrix factorization approaches. And so here are the 2 methods, SE Insight and SE Parser, that use this type of method. And the limitation here is that actually using a matrix factorization method, but also using condition, it's actually very hard to model multiple conditions. And so what they do is actually concatenate or use groups, like kind of look at the they don't separate the groups, the conditions out into distinct kind of variables. Or, people of course have also used variational autoencoder approaches, and these are also popular. But the limitation here is that the interpretability of linking genes with specific conditions is a little more challenging. And so our group also was interested in actually taking a matrix factorization approach, but hoping to address some of the limitations of being able to more directly model multiple conditions. And so the quickest review, we're going to use an unsupervised, jointly supervised, unsupervised, non-negative matrix factorization approach. But just the quickest summary of, unsupervised NMF, it's a simple idea. We have a matrix. We know that matrices, we can multiply two matrices to get that final matrix as long as we make sure the dimensions work out. But what's really valuable is if we can make the kind of two matrices that we use, that we multiply out for that big matrix, much smaller in dimension than you would otherwise need. And the idea is if these two lower dimension matrices actually can multiply out and actually reconstruct that original matrix really well, one might assume we found a really good, pretty good quality lower dimensional representation that's hopefully, thrown out some noise, and it's more interpretable because we have much fewer dimensions that we have to work with. So this is traditional unsupervised NMF. There's a lot of different methods kind of in this space. Of course, Josh here has also kind of used Liger to do lots of single cell, like kind of exciting alignment. So what we were curious about is if we know condition or batch information, can we use that to somehow guide this process of decomposition? So that's where that joint supervision comes in, right? So NMF is traditionally unsupervised, but say we're like, okay, we're kind of interested in the effect of sex or the effect of a treatment. Couldn't we like kind of steer some of these embeddings? And so that's kind of the basic setup of our method. So if we have our input data, we're going to say, let's allocate some kind of, smaller portion of that matrix. Just say that they would be good at reconstructing the kind of single cell counts that are attributed to condition 1, condition 2, or batch effect 1, batch effect 2. So we'll see that actually mathematically, we're going to be treating conditions and batches in the same way. But basically, the idea is that for batches, these are things we want to throw out later, and conditions are things that we want to look deeper into later. But mathematically, we're treating them the same way. It's just like saying, in this whole matrix of that we want to have as a decomposed representation, we're going to allocate several like dimensions or several embeddings to actually the variables of interest. So how do we do this? So we have W and H. I'm coloring kind of the embeddings or the variables that we're kind of matching. And then we're basically saying, okay, for, in this H matrix of cells to latent dimensions, we're going to basically add another kind of matrix that links that small sliver of the H matrix to some type of label. And so that's the supervision. So the labels in this case, for example, in condition 1, this could be different drug treatments. This is just like, whether or not those cells were treated with a drug or not. And then, similar for batch, it could just be, we have one row for each platform, and we're now linking basically with a kind of a third matrix, B, that's automatically learned, that will link this, that basically in a way constrains that H to better reconstruct the labels themselves. And so the conceptual difference with single cell standard integration methods is that instead of modeling cell type similarities and then trying to align them across samples, we're trying to seek to model all the other covariates that we think might be affecting things in this data set. We can still look at what's left over, which are like, the cell embeddings beyond all of the variable effects to still kind of interpret what's going on probably in the cell types. But the idea here is now, if we have this structure, we can actually immediately pull out using the H matrix, which cell types we think are associated with a particular condition. Or we can look at this W matrix and pull out kind of the signatures and be like, which conditions, which genes are associated with, which particular condition. And finally, if we look at just that unguided portion, everything without all of those conditions, we can still do batch removal. Because, we can do this in two ways, either looking at those cell type embeddings that ignores basically all of the kind of condition or batch variables, or we can actually reconstruct the count, right? We can also multiply the matrices back using only the unguided portion or actually using certain parts of the unguided and the conditions that we're interested in. But in any case, we will always kind of delete those batch embeddings. So just to give you a more illustration of what this might look like. So using we had some simulations to show kind of how this might work in like a setting where we have a ground truth. So in this simulation, basically what we actually did was generate kind of 16 different simulated cells, cell types, and then a certain portion of the cells were basically simulated as being stimulated or not based on, say, like a drug treatment. And then a certain other subset of the cells were simulated as whether or not they were kind of in a healthy or like a diseased patient. So we have two sets of conditions here, each with two labels. And what I'm just going to show you is that we could actually directly read off the simulation related embeddings in our NMS to basically see that we can reconstruct exactly kind of the same cells that we simulated, and then similar for severity. And then so that's just saying, we can identify condition associated cells, and then we actually can do more types of simulations to kind of show, what's in different levels of complexity. So in a naive case, we can say that the genes affected by these two different conditions, just random, they're different. And we compare with existing methods and show that we can, increase the level of complexity up through each condition actually affects only certain genes in certain cells, and we can still actually retrieve the actual cells better than existing methods that we are able to run. So beyond this, I mentioned that we can also still do batch removal, right? So we can actually use some of the same data sets, same single cell this time and kind of collected from real patients and samples, and compare with existing kind of very popular single cell methods. We actually find that in kind of many cases, using our embeddings, we actually can achieve really good performance, comparable or if not better to a lot of these existing methods in terms of batch correction. And beyond using the embeddings, like I mentioned, we can also reconstruct counts, right? We can actually reconstruct counts using just basically our unguided embeddings to actually also compare those reconstructed counts with count-based integration methods. So we actually can do all kinds of other kind of clever little things with this Alpine method. For example, we can actually use orthologous genes to actually integrate data across species. So you can imagine that we can treat species as a condition, right? And so we did a lot of downstream kind of analysis on this adipocyte or an adipose tissue data set that's collected in both mouse and human, cross sexes. They had multiple conditions, and we do a little bit of an analysis of that. So the takeaways I would say for our Alpine method is that it really combines the power of traditionally unsupervised machine learning methods with strategic incorporation of known sample and or data set attributes. And so we think that it really brings additional power and interpretability down to individual labels within each covariate. And so I didn't talk about this as much, but we actually can associate. So this is something that a lot of previous methods struggle with. They can, associate genes with a general label, say like it's either healthy or disease, but it's like that category. Whereas we can associate the genes themselves directly to which label you're interested in. And it's actually a really extendable modeling setup for more complex settings, depending on what we decide to treat as a condition. Or I think this kind of joint supervised, unsupervised approach, we're thinking actually really leverages the advantages of both. There's some other details I didn't, spend time talking about, but I'm happy to discuss if you have questions. As part of this, of course, you might wonder, there's multiple parameters I'm talking about. There's actually also hyperparameters. There's number of embeddings. There's supervision going on. So how do we optimize this NMS? We actually also developed an automatic optimization method. And so that basically, it's also just more user-friendly, where we actually look at, we optimize for the fact that the unguided embeddings should have little overlap with the guided embeddings. And by doing so, we can actually very quickly optimize the kind of range of hyperparameters that are involved in this method. And so we have a package already with this method that people can kind of play with. And we have some toy examples there. Cool. So now I want to kind of move on to instead of looking at single cell data within kind of single cell, we were also really interested in this question of whether we could actually leverage single cell data in combination with kind of actually population scale data. So this is kind of, I think probably many of you are already familiar with GWAS data. And the idea is again, once we were able to sequence a lot of people, one natural thought was, oh, if we can get a bunch of people with disease, a bunch of people that don't have the disease, and compare what's, the genetic variance between them, we can actually get some better assessment or understanding of genetic risk factors of the disease we're interested in, right? And so, I'll be talking a little bit more on how we're going to apply this method to Alzheimer's disease. But, there's tons of different GWAS, with Parkinson's disease, depression, schizophrenia. And I wanted to point out stuff like college education that, not a disease, but some complex traits, right? That we know there's some potential like kind of genetic background that might influence downstream kind of observation. So one big observation, though, of course, is GWAS data is super powerful, but it doesn't have inherent information regarding tissue and cell specificity. And meanwhile, we have more and more single cell data that is helping us understand that, right? And so several of these attributes that I've listed are traditionally associated with kind of neurological kind of traits. And that's because my group is also really, really interested in understanding or modeling the complexity of the brain. This is like, we talked about tissues, organs, and cells, but even within the brain, right? We know there are different brain regions are associated with different functions. Within kind of the brain, there are lots of different neuron types, and there are also support cells that aren't neuron types. So this is like the complexity just within the brain is actually kind of mind blowing. And so, people now have actually, developed kind of more and more single cell kind of atlases of the brain. The higher quality, kind of more comprehensive ones are often in mouse, but people are also starting to do this more and more in humans. And so, an interesting question is we have population scale, quantitative genetics data that's like collected over tons of people. And meanwhile, we have single cell data in just a few individuals or a few mice. But is there a way that we can integrate that, that disease variant information with these, completely unpaired expression profiles, right? It's not the same individuals that were measured in GWAS. And so we're not the first ones to think about doing this. And so a very kind of typical kind of way that people try to think about assessing these cell type trait associations is the first thing we need to do is if we have GWAS data, these are at the variant level, and we need to kind of somehow bundle them to the gene level. There's a very popular method called MAGMA that does this, which actually basically, looks at the, these common SNP variants within some window of a gene and takes into account linkage to equilibrium and assesses a disease-related Z-score basically to genes. So now we have disease kind of or trait-associated Z-scores at the gene level. We have single cell data that can also be, of course, at the gene resolutions. People have different ways of characterizing cells or cell types. So people integrate these two pieces of information in different ways. But then in the end, what we all want, what all the biologists want are statistical significance at the end for cell type trait association. So previous methods, I won't talk at length about this, but there were kind of some limitations that we thought were kind of challenging to overcome. So there was a lot of thresholding. CBRS is an example of a method that had to, you had to a priori choose like how many kind of variants or how many genes you thought would be disease associated. Or I think the MAGMA approach in the beginning also, like you can also choose a threshold for like say, top percentile or the number of genes that could characterize a cell type. And then for statistical significance, depending on the method, you might have to do kind of a lot of simulations. So you use like an MCMC based approach. This could be very, very slow, especially as you kind of increase the size of your single cell data. And so in our framework, one thing that I'm going to kind of spend a little bit of time talking about is granularity selection. And that's basically the idea that we actually realized that choosing the level of granularity in the single cell data when assessing cell type trait associations is actually really, really important. This is something that's actually been underappreciated in a lot of previous work. But what we contribute is really, we developed a specificity score. So in a lot of previous methods just use gene expression directly. And the key intuition of seismic is actually pretty simple. The idea is, especially when you're looking at something as complex and correlated as the brain, if you just look at direct expression, the signal is kind of weak. But if you can actually calculate a cell type specificity score that has a measure across every gene in a cell type that kind of highlights what's unique about that cell type, you can actually gain some power. And so we use a very kind of simple regression-based association between the magma z-score and the specificity score, and we can actually then kind of use kind of standard statistics to calculate a statistical significance for that association. One of the novel contributions of the seismic framework is what we're calling influential gene identification. So people haven't kind of done this before, but when you associate a cell type with a trait, one natural question is, why was that association called? Of course, we're interested in actually which genes and which pathways are driving that association. And the thought there is that, this could actually lead to new hypotheses, not just, say, oh, I think this particular neuron is associated with the disease, but it's this particular neuron, then this disease, and this is why. These are the genes and these are the pathways that might be kind of important for that. So before using this method on kind of real kind of data, we actually wanted to do some simulations to make sure that the p-values that we're calculating, the associations that we're finding seem to at least work well in a simulated scenario. So the first thing we wanted to do is type 1 error assessment, which is just saying we scramble the data, there's no disease signal in there. We want to make sure we're, if we report a significant p-value, it's well controlled, and we find that this is the case. So in this plot, what we're seeing is that the dashed line is basically the expected number of false positives you would get based on the p-value that you're reporting. And we see that seismic is typically conservative and it falls nicely kind of below the line. So are actually a lot of the previous methods, although we see some of them are a little bit more on the conservative side versus some of them can trend a little bit anti-conservative, but in general, everyone is trying to kind of track that dashed line. We can also do type 2 error assessment, so evaluating the power of our method. So we're again simulating data, so we're perturbing genes, so we know which genes and which cell types are the ones we've perturbed. And we can increase kind of the signal there in terms of we can make larger and larger perturbations. And so of course, I think at some very large perturbation, I think it's easier to find the signal, although some methods still can struggle depending on the correlation structure of the data. But what we see is that using the specificity score, even at low kind of fold change or expression perturbation, our seismic method is already able to pick up some signal from the data and capture the actual cell types that the system. And then I mentioned computational efficiency before. Because we're not using a simulation-based way of assessing statistical significance, we actually have a pretty good computational efficiency and we can scale up pretty well as the size of the single cell data. So we applied seismic to a bunch of GWAS studies, and this is using Tabula Muris. So it's actually a large single cell atlas collection of single cell data in many, many different tissues in the mouth. And so this is just to show you that we can actually find what typically is expected signal actually between these different 27 traits with these different tissue and cell types. So we see a lot of blood and immune kind of cell types associated with immune disorders, neurological with brain, but interestingly also some with the pancreas. We see kind of more specific non-disease traits, but measurements like HDL, LDL, or triglyceride. We actually see some associations with liver cells. So this is more like there's no full ground truth, but it is still like a sanity check that we're seeing kind of associations that we expect. Some of the additional ones that we're finding could be biologically interesting that we could kind of dig into. But I mentioned that a lot of the early motivation for this work was actually studying the brain. And so we can actually apply seismic to look at Parkinson's disease. And so, if you're familiar with Parkinson's disease, we know that dopaminergic neurons are actually the neurons that are particularly vulnerable in Parkinson's disease. And actually looking at previous methods as well as seismic, 3 out of 4 of them can successfully identify that dopaminergic neurons are kind of associated with the disease. But I mentioned earlier that we are actually very interested in exploring how can you look at kind of additional levels of granularity. And one of the motivations here is that actually neurodegenerative diseases, beyond kind of specific subclasses of neurons that are affected, we also know brain region actually is very important. So for example, in Parkinson's disease, beyond just dopaminergic neurons, we know that dopaminergic neurons in the substantia nigra are particularly vulnerable, more so than dopaminergic neurons found elsewhere. And so if you actually combine this, so in this single cell data set, we actually have brain region information as well as the different subclasses or even more detailed annotations for the neurons. we can actually look at multiple levels of granularity. And so what I'm showing you here is like previously, the result I showed you was actually for the lowest level of granularity, the subclass level, where there's actually just 14 different subclasses of neurons that were used in that data set. But we can actually kind of compose them with more and more kind of information. So we can say, look at, well, brain region is actually combined with a higher level description, so class where it's just neurons versus support cells. But we can actually combine brain region and subclass all the way down to brain region plus fine cluster, which means that we actually have 231 different cell type characterizations that we're looking at. And what we actually really thought was kind of exciting is that seismic actually performed really well at all of these different levels of granularity. And so and I think, again, highlighting the specificity of the cell types is really good at kind of maintaining kind of the characteristic features versus having them kind of compete or having the signal be too correlative and washed out at higher levels of granularity. So in addition to Parkinson's disease, a lot of, we also work a lot on Alzheimer's disease. And so if you're not familiar with Alzheimer's disease, one of the kind of interesting kind of factoids and things that people have been wrestling a lot with is that if you look at these large Alzheimer's GWAS, frequently what you actually find is microglia signal. And this is the same thing that we also find in our work. And so microglia are basically like kind of the immune support cells in the brain. And basically, overwhelmingly, these large GWAS, based on clinical diagnosis, keep finding microglia to the point that some people are like, is it really just an immune disease? And this actually, the question is kind of an interesting one because in Alzheimer's disease, just like in Parkinson's disease, there's actually also selective neuronal vulnerability, and it's selective to a particular region in the brain. So in Alzheimer's disease, it's known that neurons in the entorhinal cortex, followed by the hippocampus CA1 region, are the ones that are most vulnerable, right? And so it's kind of weird. It's like, okay, we know that there's the cell type tissue specificity, but then the GWAS keeps kind of prioritizing microglia related cell types. So what's going on here? And so what we actually did is, in addition to varying the cell type granularity, we actually were like, what if we look at smaller GWAS data? So we always go for the like big GWAS data with hundreds of thousands of people or millions of people based on clinical diagnosis. But what if we did something, look at a smaller GWAS data, but using kind of say like a pathological based, biomarker based endpoint. So this is a different GWAS study that used cerebrospinal fluid tau, measurements of tau. So a biomarker that's related to Alzheimer's disease, but it only had like 3,000 people. So much kind of in a way lower sample size, so lower powered, but much more specific, right? And so really excitingly, what we actually found is that we actually seismic was able to prioritize some of the exact neurons that we do know are vulnerable in Alzheimer's disease. So again, we thought that this was kind of interesting to highlight that sometimes, bigger isn't always better, right? So perhaps with Alzheimer's disease, only clinical diagnoses, there actually is a lot of diversity within the disease. So the pathology might be at different stages, even that's not fully correlated with the symptoms being observed. So that when you kind of bundle together like a million people or, tens or thousands of people, maybe what is conserved is the kind of immune signal. But if you kind of look at something more pathology focused, it is possible that we can actually pull out different kind of biological hypotheses. So if we go back to what I mentioned earlier, that once we have the cell type trait association, we're still interested in which genes or pathways might be driving this, right? And so we use this influential gene analysis, and this is actually again just borrowing from traditional kind of statistics. There's like a measure that it's called DF betas, and it's just actually trying to assess which observations are particularly influential in, say, a regression. And so it's kind of a very simple idea. You remove the observation, you see how much your beta, your coefficients change. If it changes a lot, you're like, okay, that was a very influential observation. And so we can do this on also our entorhinal cortex association with that GWAS, the pathology-based GWAS, and we can find several genes of interest that kind of we're following up on with a collaborator. And so takeaways here are that in seismic, we're able to integrate single cell data with GWAS to pinpoint disease relevant cell types. We're using a novel cell type specificity scoring method to link genetic risk factors to some of their phenotypes. And finally, influential gene analysis can really inform hypotheses for genes or pathways that drive disease analysis. So the last project I want to talk about is a little bit different. So we're not going to look at single cell data for this last project. And this is called Etna. And what we're actually trying to do now is diving more into this idea or thought of examining data or understanding what's going on in gene function across functions. And so we're going to take a network embedding alignment approach in this last part. So first question is, why might we be interested in doing this task of aligning, say, a protein-protein interaction network across different species? So, one of the biggest challenges, of course, in trying to learn about processes underlying human biology and diseases that we can't do all the experiments we would like or, in human, right? So we've always had to use model organisms. And so lots of important discoveries have been made in model organisms, but we also know some things transfer better, some things don't transfer as well. Is there some way we can actually leverage, computational models to help assess or prioritize which, biological processes or functions are more aligned or perhaps less so? And so this idea of knowledge transfer across species has typically relied on orthologous genes, right? So the idea has always been you have the same quote unquote genes in different species. And we know that, there can be multiple such genes. But there's some limitations here, of course, as well, because we know only a limited number of genes have orthologs, and orthologs are actually imperfect. So, these orthologous genes are typically assessed by sequence similarity, but sequence similarity does not guarantee functional similarity. In fact, sometimes you can actually have re-ranking, where sometimes the thing that's most sequence similar is not the most functionally similar, and sorted different. So, protein interaction networks, meanwhile, have been really useful as like a snapshot of protein function, right? We know that proteins work together to carry out functional roles. I mentioned in the beginning. Functions are also often shared between physically interacting proteins. So we know that these have been widely used for identify disease genes, pathways, inform drug target selection. There's lots of downstream kind of paths that have been really valuable. So here, the natural idea is, can we actually align protein-protein interaction networks and not just use sequence similarity between genes to prioritize quote-unquote functionally similar genes? So network alignment, if we don't consider the orthologous genes at all, first starts with considering structural similarity. So here, the toy example is that if we had a human PPI and a mouse PPI, we can look for a kind of which areas, so structurally speaking, which parts of the network are more or less similar, right? So this problem has actually been very, very well studied, starting from IsoRank in 2008 that took this approach of thinking about, so it's like a PageRank-based approach for aligning two networks. So there's actually, some methods that only consider network topology that I folded here. Meanwhile, a lot of the other ones actually consider similar problem to us of how to combine structural similarity with sequence similarity together. The challenging part, of course, is that there might be actually inconsistencies between kind of what's sequence similar and what's structurally similar. And so one of the biggest challenges a lot of previous methods had was actually they usually just give you a tuning parameter. And so you can just be like, do I want to be more on the sequence similar side, or do I want to be more on the structural similarity side? And so the perspective here is that these are kind of pit against each other, right? They're kind of on opposing scales, when biologically that's kind of also not necessarily what makes sense under the hood. And so recently, semi-recently at this point, so in 2019, there was another method called Monk that was thinking about formulating this problem as thinking about a joint embedding between these two kind of species versus saying, okay, so the idea is that it could be a nonlinear combination, right? It doesn't have to be like, tune this one up and tune that one down. And it could be different for different genes or different proteins. So we set out to develop a method where we wanted to be able to do a bidirectional mapping between species, because as we'll see kind of downstream when I show a comparison, one of the challenges of Monk is that it's actually a unidirectional mapping. So what this means is that when you tell it that you're mapping human to mouse versus when you're mapping mouse to human, you actually can get very different results, which again, not fully intuitive, because it's like, you can actually get very, very different mapping. We wanted to be able to get, of course, a lower dimensional embedding. Like I mentioned in my kind of first project, we care about low dimensional embeddings because we think that they're both more efficient, but also more importantly, often better representations, more interpretable representations of the original data. We want to be able to capture nonlinear relationships between topological structure and sequence similarity. And we wanted to handle the sparsity of PPI networks. And by sparsity, I mean that PPI networks, they're really valuable, but they're still largely incomplete, maybe except for in East. Basically, this involves a lot of high throughput screening. So when you have an edge that's missing in the PPI network, it could mean either the two proteins don't interact, or it could mean that those two proteins have not been studied kind of as a pair together. So here, the project that I'm going to be talking about, Etna, they actually break this down into 3 steps. And so the first step is what we're going to do is create individual species network embeddings. And I'm going to talk a little bit more about how we set this up. And because it's not necessarily completely intuitive, the second step is once we have an individual species embedding, what we actually kind of take is actually inspiration from natural language processing to do a language translation or a network translation task where we can swap the encoders and decoders from the previous step. And we basically, the idea is that we're going to be cross-training these 2 networks to make sure those embeddings become more and more, in a similar space. And once we've declared victory that we've had these 2 embeddings in a similar space, we can actually then, analyze scores between, proteins from each of the corresponding species, right? We can, of course, also look at within species, so we can actually then assess how similar are these 2 proteins in this, joint embedding space. So diving more into this first step, the individual species network embedding, one natural way, and a lot of people have been using in, using neural networks is to actually just make an autoencoder. And so autoencoders are really great for finding low dimensional embedding. But the big question is, we start with a network. So how would you throw the network into an autoencoder to find a lower dimensional embedding? Or more specifically, what are we going to optimize for that output to match, right? So we have an adjacency matrix starting out for a network. And so, of course, naturally, we can actually want to reconstruct the edges you find or the actual full adjacency matrix of the network. One kind of important thing that we kind of account for in our reconstruction loss here is that we only want to pinpoint and care about the fact that we have a positive edge. We don't want to say we're focused on reconstructing all the negative or the zeros because, as I mentioned before, we know PPIs are sparse. So as part of our local reconstruction loss, we focus on how well we're able to reconstruct observed edges in the local reconstruction, the local kind of neighborhood of a particular approach. So it's basically, if you have an edge with somebody else, we hope that after the, passing you through the autoencoder, you're actually also like the, 2 proteins are close together in the embedding space. But beyond that, we actually know that one of the valuable kind of aspects of a protein interaction network is not just direct neighbors, it's actually distal relationships, right? So it's like the friend of a friend is still also interesting and different than say ones that are completely kind of separated from each other. And so we use a closed form estimate of the speed block measure, and I'll just give you some intuition for how that works. And the idea is to provide similarities for nodes that may not actually have a direct connection with each other. So how does this look like? So people have done a lot of what people call random walks on networks and shown that this is actually a really, nice way to summarize farther relationships between nodes that aren't just, do I have a direct neighbor or not? And the idea is you start from each node, you keep walking some, path down through the network. How often do you reach these other nodes, right? It's like if you reach them frequently, then you're kind of closer in network space. And so we combine kind of basically this closed form estimate of basically this type of a random walk together with the fact that we want to also prioritize if you do have a direct neighbor. And so that kind of these 2 components constitute our reconstruction loss for individual species networks. So now what do we do if we have 2 species, we have mouse, we have human, we are hopefully thinking that we have good quality embedding for both of these networks. So we use this cross alignment idea. So I already mentioned basically we were inspired from NLP, which in the early days when they were, or medium early days in NLP, when they were doing language translation, they basically thought about, if you kind of, it's a very intuitive idea. If you have, say an English autoencoder, and you have a French autoencoder, you can swap the encoder of the English language and say you're decoding it in French. So it's like you input is English, output is French. And so in this case, we're thinking about for us, we can think of proteins as kind of words and orthologous proteins as words that are more likely to have similar meaning. We're not considering that they have to be identical, just in the sense that in natural language processing, when they were doing the language translation task, it's not that the words are going to be identical either. You can actually have these these words are more similar. They're often the similar things, but we make sure that the cross training is focused only on those anchors, so to call them. So we're not saying that, so basically these ones guide the embedding, and the idea is we iterate between the individual species network embeddings and the cross training. We iterate between them, updating only the encoders each time, because we want the decoders to actually be fixed. So we're saying that hopefully now the encoders are better and better at making sure that those two embeddings are kind of interpretable in relation to each other. So once we do that, we are then able to kind of say, okay, let's assume that that worked really well. So that means that the mice and the human, that this embedding representation of their networks is kind of in the same space. So we can now just do simple kind of in the embedding space, we can now calculate cosine similarity or any pairwise similarity or distance metric between any pair of genes, whether it's in the same species or between species. So of course, before I go on, everybody will want to know, how do we know, can we have an orthogonal evaluation besides the loss function that we ourselves define to see if these embeddings are any good, right? So for our evaluation, we use one of our favorite resources, gene ontology. So of course, we know that for gene ontology, lots of kind of scientists have gone through an annotated gene function based on published literature. There are evidence codes associated with those gene associations, so we can make sure that we're only going to pull out genes that are associated with functions based on some experimental validation. And then what's really cool for our particular evaluation is that people have done this not in a species-specific way. So in gene ontology, within these Go terms, we have genes from multiple species. So that's actually a great kind of proxy for, genes from different species, and if it's the same Go term, we can think of them as at least having some aspects of function that are similar, right? And so the one thing to note here is that there is actually a many-to-many mapping between genes and Go terms, right? Typically, a lot of genes have multiple Go terms annotated to them. And so if we think about this, we actually can think about, say a human gene and a mouse gene. Both of them have multiple Go terms annotated to them. And we can actually then calculate a Jaccard index, or basically a measure of how similar their overlap versus their total pool of terms are. And so that kind of gives us a score. For every pair of human genes and mouse genes, we'll have some score that represents, at least based on our current knowledge, how functionally similar those two genes are. So what we can do is now compare that score that we get with those cosine similarities that we got after we did our joint embedding. And so what we find is that consistently across methods, if we compare, if we've now aligned mouse to human, or yeast to human, we've also done this with fly and worm to human, anything with a reasonably kind of large PPI network, our ETNA method actually outperforms previous kind of the main kind of network alignment methods that don't use, that Monk uses an embedding approach, but isorank and Hubble align, two classic methods that don't use an embedding-based approach. One other thought we had was actually, how does cross-training actually affect the overall quality of these embeddings, right? So I just said that we make an individual network embedding. Now we're doing this cross-training, going back and forth. Was it actually bad? Did it make those embeddings worse in order to constrain them to be more similar to, say a human and a mouse or a human and a yeast, right? And so what we actually did was actually, so if I show you here, is we can actually look, my previous evaluation was actually just looking at human and mouse genes that were annotated to the same Go term. We can, of course, look at human and human genes annotated to the same Go term, right? And say, those two genes should also be functionally similar. So what I'm showing you kind of 3 bars here is red is ETNA. So again, this is actually ETNA with the cross-training. We also can take out the cross-training step. And we see that actually cross-training actually is improving the embedding even just for human genes. So that was really exciting. And then we actually did an additional test where, I mentioned that we thought we wanted to have both the local and the global reconstruction losses in that individual network embedding. So we can also remove that global one, right? So we're just only going to look at direct edges and not the distal edges. And we see that that actually makes it a huge impact. So it's really important to consider that those distal relationships between genes when we're doing this individual network embedding. And so here, what we're seeing is that, not only is cross-training not making the embeddings worse, it actually can make them better. And the rationale for that we believe is that you're borrowing information from the other organism, right? So here I showed the quality of the human embedding after aligning with yeast. And in this right figure, what I'm showing is the quality of the yeast embeddings after aligning with the human. And we see that actually, of course, yeast, there's much more data, the network is much smaller. And so overall, the predictions are all better, but the improvement after cross-training is actually smaller. So our kind of interpretation after seeing this is that the human was able to borrow more information from the yeast, where the human network is actually more incomplete. And the yeast still can benefit a little bit, but less so. One kind of just last example of what we can do is if we have embeddings, we can actually use those embeddings as input into downstream machine learning methods. So that's something that traditional network alignment methods without kind of an embedding space, you can't do. And so one task that we thought would actually be interesting to evaluate is genetic interactions. And so genetic interactions, just a quick summary, is like people do these systematic knockouts where, basically if you have gene A and gene B, you see a viable organism, typically yeast. You would knock out one gene at a time, the organism is fine. You knock out both, it's called synthetic lethality, the yeast die. And so the idea is that something shows that A and B, they might not be physically interacting, but there's some relationship between them, right? And so we can actually set up our method where we can actually say, okay, let's just sum the embedding of gene A and gene B and input that into a supervised machine learning classifier. So in our case, we just use an SVM, very simple. And now we have, for a given pair, we have a feature that represents them. And we can just say, okay, now we give you a gold standard, here are some examples of synthetic lethal gene pairs and some not, predict more from. And so here's actually a table of the results. Again, I mentioned in passing earlier that this monk method is not bidirectional, which means that there are two sets of results for depending on which source and which target organism we use. And the abbreviations here are both yeast types. So SCE is Cerevisiae, common yeast, and then SPO is Pombi, a different type of yeast where people and both organisms have done a lot of genetic interaction experiments. Cerevisiae, SCE is the one where that has the most data. And so we'll see that basically what I'm showing you is like, you can do a very simple experiment first, where we're taking basically a combination of gene pairs from both SCE and SPO that have been declared to have genetic interactions and predict for a single organism. These pairs are held out. Not only are the pairs held out, we actually make sure that there's no genes that are shared in the holdout training set, or the training set. So that means that, we of course lose some gene pairs, ones that span the two, but this ensures that there's not, cheating basically by our method by kind of peeking at any of the genes that are in the holdout set. And so, we see that our method performs kind of well in all these cases. We can also do the harder task of training on only a single organism and predicting in the other one. And so there are dashes there to show that basically because in the previous kind of case, when we used two organisms, we actually subsampled SCE to be as small as SPO. If we use the full SCE kind of set, monk was actually too slow for us to run. It wouldn't converge. And so we weren't able to run that experiment. But more excitingly, we can also do this for human. So human, there's been some genetic interaction experiments as well. And the idea is we can actually then use our method to transfer interactions between yeast and human. So just to wrap up, I know I'm running low on time. We actually, once we do this, we can actually do some other fun downstream interpretations where we can make we have now mouse genes and human genes all in the same space. We can do some clustering on them and actually look at basically gene set enrichments in this space for diverse like mouse phenotypes, as well as human, say, Omenn genes, GO term, and GWAS, disease-related genes. And so then we can kind of start to get a better map of like what's interesting kind of cross-species relationships that might be captured in the protein-protein interaction network. The takeaways here is that we're able to find embeddings that preserve both within and between species gene functional relationships. It provides a new way to unravel gene relationships that are hard to detect experimentally. And finally, we can actually use this type of method to reveal shared mechanisms of fundamental biological processes captured in model organisms and hopefully human disease pathology. And there's kind of some, interesting downstream applications that we hope to apply them. So yeah, these are the three approaches that I talked about today. And I think throughout, I think there were kind of the students that worked on it, but in this is my lab. Thank you all for listening.
Where will it take from?
Hi, thank you for your talk. I'm really interested in the Etna project you talked about it. I'm just wondering if you could say a little bit about how you trained the prepared autoencoders. And it sounded like you first trained the autoencoders and then you did the contrast over the learning the shared and embedding space separately. Or second, I'm just curious if you could say a bit why you did it in that order.
Well, so we actually trained so I presented it this way, but we defined the individual network kind of embeddings, but it's actually a cross training. So we actually are updating the encoders. Once we actually have trained those reasonably well, we're now updating the encoders over and over again, but I'm not updating the decoders of the network. And the idea is there is if you allow both things to vary, you're actually not making sure that the embedding, which is like the bottleneck in the middle, is actually similar, right?
Why is it better to fix the decoder rather than to first learn the shared embedding space and then train the decoder separately?
So we're fixing the encoder, not fixing the decoder. So we fixed the encoder.
Thank you. I did not hear. Thank you.
Yes. Yeah. Sorry, no, we're fixing the decoder because we don't want to update so we want to make sure, basically we're updating the encoder, we fix the decoder. So the idea is that when you decode, if you allow that to change, then you can't make sure that the shared embedding is actually kind of similar, right? Because now your decoder can actually change how it, decodes the embedding.
This is a good part. These are very cool projects. I have a technical question regarding the first one, Alpine.
Yeah.
That you're trying to disentangle the latent factors that are corresponding to different conditions or batches, or and at the same time there are also some shared factors that kind of like underlying shared biology across different conditions, different conditions. I'm curious that whether, I guess, imagine a case where if you don't separate out some factors, just like strategically them for a certain condition, the remaining one will encode some of the batches at or condition. And I wonder, like, even though, like, right now you're definitely some specific factors part of the system, I wonder whether you need to still have some regularization term or have some guarantee that the remaining one is truly shared. It does not encode anything, does not have any correlation or dependency with the designated condition or that person.
Yeah, so that's a great question. I think so I think I mentioned, I can pull up so I think I mentioned that we have an optimization kind of procedure, and the optimization method is actually trying to basically make sure that basically whatever is left in that unguided portion does not basically form clusters that have significant information captured in the any of the conditions or batch effect variables that were provided. So the caveat there, of course, is that if a variable was not provided, that actually can dictate, that's say a batch variable that was unreported, that would be probably in the kind of unguided portion. Of course, we could use kind of like a surrogate variable analysis kind of type method where we could imagine that some of those unguided variables, we could automatically learn whether or not they're actually also batch related. And then I want to show basically, like you actually have suggested, I glossed over this, but we do have a lot of regularization actually in place. So, so basically, so this is like, I skipped over this slide, but basically the way we actually set up our NMF. So we have the standard NMF reconstruction loss, and then the prediction loss, and then we have this last term that is actually both elastic net regularization and trying to make sure that the embeddings are more orthogonal. So that in self is actually making sure that the embeddings themselves don't actually overlap a lot. And so all of these hyperparameters are actually also tuned as part of that automatic hyperparameter optimization.
In the second,
Yeah.
GWAS, the single cell. So one of the things that is interesting to me to think about is how many of the processes that underlie this are actually going to be very because this method and what it can do, I've experienced some higher right, that we going to do such a discussion, and this is in the work time, going to bring to that. So can you tell me a little bit about, you know, this kind of, what happens to the really
Yeah, so I think that inherently, just as you said, the method is going to only pick up the cell type specific portions, aspects of the disease that are kind of related. So the method, I think the way it's set up, there's no way I think we could actually glean kind of external factors beyond that. I think that interesting similar approaches could be taken if we have single cell atlases for like say DNA methylation or other types of modalities that could capture other types of relationships. But I think inherently this is going to be focused on, say, you know, what's inherently different about the cell types, which I think is still, I think the motivation there is some aspect of the disease that is still driven by cell type specificity. And so that method seismic is only gonna be focused on that aspect. I think for other types of contributions to disease, that would, you know, I think require other types of data integrated with or other diseases. I think actually I was interested recently to hear about like kind of rare variants or other kinds of similar quantitative genetics kind of you know, findings people are having that can summarize disease gene association.
Is there any question from online?
Yeah. Yes.
This question is, how is the community between government because from what I understand, you both convert and very into the binary processes and factorize and then the idea of the community
Yeah, so it's not related to the number of genes, but it is related to the cell. So basically it's connected through H. So I think, sorry if this wasn't really clear, but that H, that's the BH, is the same H in the big H, but it's only a sliver of that H. So it's not all of the H, but we've said like, okay, you know, first 4 embeddings, they're assigned to that condition. So because that H is what connects them. Because now the B is connected to H, but H and W are connected. So W and H are jointly trying to establish that reconstruction loss. And B and H are trying to basically establish. So we basically figure out the updates. You just update W, you update H, and you update B, right? And yeah. No. B and W don't see each other in the loss function, but H links them. Yes. Exactly. Yes. So basically, our kind of hypothesis, what we actually often show is you can take those unguided embeddings, and that's supposed to be cell type, but you can actually pick and choose whichever other embeddings you want, right? Of course, never the batch ones, hopefully, but then you're picking like a condition or a set of conditions at a time. You can actually visualize those together, or you can actually multiply those out with the corresponding W's to get counts. the idea is that those are the counts that you would have maybe gotten if you didn't have a batch effect, or you only were looking at these conditions and not those other conditions.
There are any other questions? Okay, thank you very much for this very engaging discussion, and let's Vicky for one more talk.
Okay, thank you.